All right. Welcome, everybody. We have a good group today as well. Thank you all for joining us for our CSU webinar series. I have the utmost pleasure of introducing Dr. Jean Davidson, who is currently on sabbatical in Iceland and at some ungodly hour of the night. But then again, it's always night there, right? So
That's all the same.
Dr. Davidson got her PhD at UNC, is that right?
Yep.
In Chapel Hill. And then she followed it up with a postdoc at Stanford, after which she actually worked as a genomic scientist for a little bit before starting her faculty position at Cal Poly SLO, which I had the very pleasure of visiting last year. And I had such a great time, such a beautiful campus. And if you've not been to San Luis Obispo and Paso Robles and all that area, I highly recommend that you go. And Dr. Davidson is going to tell us a little bit about her sabbatical this semester. So without much further ado, take it away.
All right. Oh, I have some of my students here. Thanks, guys. It's cute to see the little chat. Thank you, everyone. It's always lovely to see you. We were just nerding out. One of my favorite things about science, and maybe especially bioinformatics, is the community. I feel like we all are just dorks who love it, and like it is such a wonderful community. And I really want to thank Scott for putting together this seminar. I look forward to it every year, and I appreciate being able to join in from Northern Iceland on my sabbatical. And I'm going to talk to you about what I'm doing here, how I got here, and a little bit about a cool science project that I am working on here. Okay. Da-da-da. So, right, as was introduced, I am an associate as of a month ago, professor at Cal Poly San Luis Obispo, brought on to teach genomics and bioinformatics, which I just spelled wrong there. Oof. But I am currently a Fulbright scholar at the University of Akureyri in Iceland. And if you have any thoughts or questions, please my email here. I'd love to chat about anything that I have in this talk. So I'm going to tell you a bit about my background, how I got here, because it is a unique story. What I really pride myself in doing at Cal Poly, why I ended up in Northern Iceland. What I'm doing here is teaching and research. So we're going to go over some of that perspective, and then I will talk about where I see this all wrapping together. So as I was putting together, I was like, did I ever think I'd be at a bioinformatics seminar, on a bioinformatics sabbatical? And, and we were just talking about sabbatical makes you very like retrospective and what are we doing here? And I'm falling into all the cliches, but putting this talk together, I did my first just awful RNA-seq experiment in graduate school at the end. So 2011, 12. But I knew, even though it went terribly, I was hooked by this idea that computation, technology, biology, curiosity could intersect in really interesting ways and answer some just these big, exciting questions. I will say I am a practitioner and a champion of bioinformatics. I am not writing my own algorithms. I am very much using existing tools, optimizing tools for my own biological questions. I love to collaborate. That's my favorite part of bioinformatics. And I really love to encourage students and colleagues to think about how technology can help advance their science. So how did I get into this perspective as a scientific career? Oops, I went the wrong way. As was mentioned, right, I came out of UC Davis actually for my undergrad. I was a plant biochemist. I just liked science. I didn't really know what I wanted to do with it. It wasn't until I went and worked at UC San Francisco that I got my first staff scientist job, and we were working on cystic fibrosis. And I was like, wait, all these genes that I love and cells that I love could help people. That's amazing. I need to do that. Worked there for a few years and then went to University of North Carolina for my PhD. Did a really fundamental genetics, cancer, cell bio PhD. Started my postdoc at Stanford in hopes of, I got the taste of sequencing and genomics. I went there with the full intent of being a computational biologist, but that was too big of a leap. My first postdoc was in epigenetics, and I was a really great molecular biologist who would send off for sequencing, and the bioinformatics team would take it over. And that was great. That was the right role for me at the time, but it wasn't in my heart what I wanted to do. So I left my postdoc kind of early, only because it wasn't the right path for me. And I became a staff scientist, and that was absolutely the right path for me. I got to work on all sorts of really interesting big data consortium science projects, things like ENCODE and the Undiagnosed Disease Network. I got to bring my biology in and my molecular biology and in trade, I got to learn data science and data wrangling and as much programming as I know from those experiences. So just a little encouragement that the path is what your path should make it to be. So I was happily a staff scientist at Stanford, and but feeling a little, what's next? Do I want to be a staff scientist forever? I think I'm kind of ready to maybe try something else out. And luckily, Cal Poly San Luis Obispo was hiring a genomics professor, and I grew up in San Luis Obispo. I should mention that. So I had kind of an insider's perspective onto this unique place. So San Luis, you're all from California. It's one of our 23 CSUs. It's big. It's lovely. It's a PUI. It's a strong ag and engineering background. A lot of like wine is huge. Paso Robles is right there. Ocean's right there. Awesome. But when I got brought on, it was because of this really big donation that they were going to build their math and sciences. And so the cheesy phrase like keeping up with the Joneses. I feel like they felt like they needed a genomics. They saw the field. They really wanted to buy me a sequencer. They knew that this was an important thing that students should learn and be engaging with. But I knew San Luis well enough to say, I'm not going to be sequencing human genomes here. I'm not going to be doing things like I was doing at Stanford here. But what I can do is that enthusiastic champion of bioinformatics and collaboration and connection. And here we are kind of isolated in the center. Can we train up our students and really empower them to do the things they can do well at Cal Poly and not pretend like we're going to be a Stanford light, because we're not. We can do better than that at Cal Poly. And it worked out. It was a swinging and it worked out. I was lucky to start this job in 2018. And a couple of years ago, I don't think it was last year even, I should probably get rid of it, but we made the like Spotify Wrapped. And that was like the only way people have ever heard of San Luis if they're not from California. But it was on Twitter and everything. We were very excited. So when I got to Cal Poly, I had my main interests. I was going to teach math, biology, and genetics. I like intro classes. I was going to teach some non-major classes, which has been really interesting. My bread and butter, though, I teach bioinformatics, genomics, gotten into some data science classes. I do research as best I can at this with the confines of precision medicine, molecular diagnostics, under this collaborative, a lot of open source tools, a lot of open source data sharing. I run my lab collaboratively with a great colleague, Paul Anderson. We support, you've been studying this oyster for 10 years. Let's help sequence that oyster genome, et cetera. I've helped build our bioinformatics minor, which I'm really proud of. It's very applied and has this really fantastic capstone experience. We're now about to launch our very first data science major, which I'm thrilled about, where a track is on health informatics. So I've been getting more into that field. And we're in the works slowly, but hopefully things are moving to start a clinical lab scientist program, a genetic counseling program. My big goal is, the students are ambitious and smart and want to save the world. Let's give them every opportunity to do that. Let's make sure the things that we're teaching them get them jobs, get them the jobs in the fields that they want, that they could stay in San Luis maybe and not have to move to Bay Area or LA. So that's my curricular goals or in service goals. And then I just love it. I love the CSU bioinformatics community, big member of the CSU biotech. I love thinking about industry collaborations. It's not a bad word. Industry sponsoring our labs, sponsoring our research is not a bad thing. Internships are great, and really with an eye towards improving healthcare in resource limited communities. San Luis is a very wealthy part of the world. It also has a ton of health disparities, and I think our students are uniquely primed to really address some of those disparities. So I did all that for 5 years, well enough that they gave me tenure, and hooray. And so I applied for tenure, thank you, and got it. Same year I applied for sabbatical. So I arrived in Achieri in August. We're staying through December. I am teaching bioinformatics here and doing genomics collaborative research and really trying to establish some ongoing collaborations between San Luis and this university. And it is thanks entirely to the Fulbright program. And this was yesterday when we thought there was a lot of snow and we were really excited, though my very California husband was still wearing shorts and it was like 27 degrees or something. But I want to just give a brief plug to the Fulbright program, mostly because I heard about it listening to someone else's bioinformatics talk, and it greatly changed the trajectory of my life. And so I'm trying to pay it forward. I was at a teaching bioinformatics at Reed University the summer before, and someone mentioned their experience, and I'd never thought of that before. And whoever's in the audience, I catered it to both sides. The Fulbright program, we often think about it for students, and it is. It's a really wonderful opportunity. Students should look into Fulbright programs for a semester, a year, whatever you want to do. It's a really great international exchange sponsored by our State Department. You get a lot of support from the United States with your visas, and you don't have to be a citizen. Like, it's a really wonderful program from students. It's also great students for or great experience for faculty and staff. And so I went through the Fulbright Scholars Program, but I'm going to give you my secret sauce to this. A lot of time people think Fulbrights are, I'm going to write my research plan, kind of like a grant. Like, I'm going to write a grant, and just the United States and India think it's really important that I do this. And that's great. But it's really hard. It's like writing an R01 to science, or like, I'm just writing a grant to the world. And so unless you have, like, I've been working with these people for 10 years and I'm from there, or this one fish lives in this one pond there, they're very, very competitive. And as a bioinformatician, I objectively do not have that. I just said, I want to collaborate with whomever. I want to march around and do fun science that happens to be nearby. And so I was like, I don't think I can get a Fulbright because I cannot in good conscience say, I have to go here to work with this person, at least in my experience, totally in my own experience. It just didn't, I was like, not for me. There are some 20% of Fulbrights that are like job ads where you go browse postings that the world puts out. And that was a goal. I literally typed bioinformatics in a search bar, and it brought up not a ton, but like 5 or 6 posts that were, and you could read through them. And this person's on sabbatical, and this is a new university, and this is a new program. And you kind of went through, and there was 2 that I felt qualified for. One was in Spain, and one was this one in Northern Iceland. And we will forever wonder why I picked this one, but I think it was in my heart. I think it was the right one. I applied to the Northern Iceland one, and you wait and wait and wait. And I think in March of this year, you get a letter and they say, congrats, you're going. Here's your visas. And they really do a nice job supporting you along the way. So I'll just put it out there. If it's on your radar, to don't shy away from those RFP style ones. We're talented. And it was like, do you want to teach bioinformatics and do research and collaborate? And I was like, yeah, I do. I do. And I don't really care. Again, I've always said I don't care where I am. Let's put that truly to the test. Do I not care where I am? So I did get to chat with a few of my colleagues before I started. I met with them on Zoom. I wrote 8 letters. Like, does this really feel like a good fit? And that was hugely helpful. So when I applied, they actually knew me already. And so they wrote a letter of support for my application, which that felt really great. It did not feel like such a rodeo of just throwing my name in a hat. And then I had to go look on a map at where Ankeri is. So Reykjavik is down here. It is the largest city. The whole country population is 400,000, which is the city of Oakland. The state of North Dakota. Like, it is a small population. It is a small country. But most people, about 100,000, 200,000 people are in Reykjavik. And then everyone else is spread out around this island. Here at Ankeri, we're the second biggest town, capital of the North, we're called. And the town is about 15,000 people. So it goes down very small. And then there's towns out here that are like 10 people. There are glaciers, there are fjords, there was a volcano down here. It's a very exciting place to go. And there's also this lovely university. It is a free public university, supported by the government. There's one in Reykjavik, there's one in Ankeri. The one up here is supposed to be a bit of the like biotech, ag, engineering one. So I was like, that's like a San Luis. It's not near the big city. It's up here. It's got this like applied focus. Like I can do this. I can teach bioinformatics there. And I was thrilled to meet with all of these colleagues. Odar and Christian, they were teaching the class previously, and this is work we're all the research we're all working on together. Margaret is our chair and also involved in the research, and then Natalia is actually was doing her PhD here, and a lot of the research I'll talk about. She's just left to start a postdoc in Sweden, but I'm kind of finishing up some of her work here. So huge credit. All the science is going to be really built on the shoulders of these scientists. My sabbatical goals, or what I signed up to do, here's what I'm doing. I'm teaching an upper division bioinformatics course. It's 3rd and 4th years, undergraduates. Some masters, there's a major called biotechnology and there's a major called pre-health. It is a small university. It's about 2,000 people on a good day when it's not snowing. And so the majors are pretty broad, but I have about 30 students in my class. Research, and I said I'll come and I'll bring genomics and bioinformatics we're not going to have sequencers, we're not going to sequence human genomes, so we can do exciting innovative things with bioinformatics on the existing projects that you have going. And then wouldn't it be great to keep this up? Let's establish some collaborations. We have these similar veins and styles. Can we keep up these collaborations going forward? Maybe we can switch some faculty back and forth, some students back and forth. So those are my goals. I just want to touch on the teaching for a second, just because we were talking about, bioinformatics is luckily kind of solidifying into a field where the courses are kind of coming together. There's some textbooks. I feel like there's some nice, so I split it up into 4 modules. We started just with the basics and file formats and how to sequence, quality control. We spent a couple weeks, I generously called algorithms, but we got into like the fundamentals. We talked about day hop and pairwise and assembly, and really spent time looking at the algorithms, not writing them from scratch, but can we just appreciate them, what decisions were made, did some phylogenies, really got into pipelines. A big chunk of time, and we're right here, actually, we just did our metagenomics today, is in the applications of this. How could you use this? And so we have really practical exercises in all of these. I'll give you some data in all of why these are really cool areas of science, and then I'll give you some data and you will go off and do it. And then we'll end, right? Big picture, how this is going forward, what we should all be worried about or not worried about or dream big about. I want to talk about the tools then for just a second. There's a lot out there, and I had to make some choices when I was here. I'm away from my servers. I could have VPN'd in. I don't know. I wanted to keep things like as simple as I could. And I don't know, it's going well. And I'm going to say the word galaxy, which I know raises some eyebrows in my bioinformatics community. It's been awesome here. So we meet twice a week. We have like 2 hours twice a week. So I often do kind of a 45-minute lecture with background and theory and as much as I feel like they need. And then we really get into the applied and exercises. So I'm going to echo, Galaxy did a phenomenal job just putting data sets out there. That's if you want a paired end FASTQ of E. coli that has an interesting variant in it, you can find it and why and what paper it came from and why you might be interested in it at Galaxy, at these tutorials. I've customized some of them. I've done some of them straight out the box. I've skipped some parts, but I had everyone set up their own Galaxy instance. We've done a few of these Galaxy trainings through the way. I give them their brand new FASTQs and they do it on their take-home homeworks. And so I get them out of just the tutorials. It's been great. We always go look at the code. We see how you can go access the code. But again, in this, do you want to just try using it? I've been a big fan. We also have used luckily there's now finally a web app for IGV, so you don't have to download it. So we actually get in there and look at our BAMs and look at what those BAMs and those variants look like. I've done much more with UCSC than I've ever done, building our custom tracks and uploading our own data and manipulating our own data. I mean, I feel like I'm talking to my old friends, but they've really come through. And then we're doing a lot going and getting raw data, going to EMBL, going to GenBank, RefSeq, Go get your own data, bring it through Galaxy, put it on the UCSC Genome Browser. Um, it's been working out really well. And then I want to give one shout out to sandbox.bio. I don't know who put that together, but they are phenomenal. If you do, I always struggle with, but I'm not doing Unix, I'm not doing command line, I'm not teaching them programming. It's just such a big ask in a bioinformatics class. Sandbox.bio has Wrangle a fast queue in this like virtual terminal. So the files are there in nice little, and it has the tutorial on the side. If you ever wanted to improve your wrangling skills and file data management skills, Sandbox, and why you should want to do that with bioinformatics, Sandbox has done a really user-friendly introduction to that. So these are my tools. It's been working so far. Okay, but let's talk about the research. So Iceland is fascinating. It is this small little island. It is close to Greenland, close to Norway, close to Sweden. It's a Nordic country, but it's very much its own thing. It is, in terms of geographically isolated, culturally isolated a bit. They speak Icelandic proudly. They speak English if you make them, but they speak Icelandic first and foremost. They are very proud of their heritage. Decode is here, which has been keeping track of people's pedigrees since there was the idea of pedigrees in people. And it was really fun to go visit Decode and what they're coming up with in their GWAS. And then ecologically, I think it's one of the most interesting places. I mean, the jungles we love and stuff, but if you wanted to get like a geographical island, right? So isolated, gene flow is really constricted, that has extreme climates, glaciers, 22 hours of sunlight to 22 hours of darkness, next to lava fields and water that is too hot to touch. Like it is all here and within an hour drive of each other. So again, we're up here in Akiari, and the colleagues that I were working with, here's my story. They were interested in lichen. Moss is everywhere here. There's not grass really. There is grass, but there is moss beautifully everywhere here. So, and there are lichen everywhere here. There's just these big meadows. This is all moss and lichen kind of cohabitating. And there's it's just beautiful landscape. Lichen are a stable symbiosis between fungi and bacteria, and of a cyanobacteria. Typically is like what you learn in 6th grade. The cyanobacteria does the photosynthesis. The lichen is the like house for it. It's this beautiful relationship. Lichen has over 800 species, some just endemic here, some seen all over the world. It's a really interesting organism. It's this, commensal organism. It's a really great indicator species. It's really sensitive to pollution and to soil qualities and air qualities and stuff. But it can also live in really sulfur-rich regions on volcanoes. It can live in really dark regions. It can in really hot regions. And so it does well here. It has adapted to a lot of these little environmental niches. So it has this very diverse biology, very interesting metabolites. It's able to put up, its genome has co-evolved to really overcome some tough challenges. So these lovely colleagues that I mentioned before were interested in who was like living in these different bacteria, in these different lichens and different mosses and different ferns, did this wonderful study where they went all over the country and sampled lichens and mosses and ferns, mostly from the north and some from the east fjords. This is just a map of where the sampling happened, and made me like, did I miss my calling as an ecologist to go tramp around in the field and collect the samples? But I've made my choices. They brought back the samples, and they had this lovely paper that found Pseudomonas syringae in lichen in this one genus Peltigera. So they went and sampled, and they did some plating, they did PCR, and they were actually able to identify via barcode sequencing Pseudomonas syringae in these lichens. They looked at 10 different lichen genera, and only Peltigera had them. They also found them in the moss and in the ferns. And this paper really gets into the genetic differences between the moss and the ferns, and like what is the genetic evolution of the syringae in there. But syringe should, I don't know if you took a plant biology class, we were plant biochemists back in the day, it should raise some eyebrows because Pseudomonas syringe is best studied as a major plant pathogen. It is a gram-negative rod-shaped bacteria. It has 50 known pathivars, meaning it has a lot of diversity even within this species, and a lot of diversity in its pathogenicity. Some are totally innocuous, some are quite aggressively pathogenic. There's anytime you study like type 1, type 2 responses in plants and that arms race between host and pathogen, this is a key example of that. So it raised an interesting question of what is it doing in lichen? Is it there on accident? Is it there in a pathogenic way? What is it doing in these lichen? Because they seem fine, these lichen. So they found that this lichen was only in this genus Peltigera, and Peltigera, here's a few pictures. Then they again linked to a paper kind of getting into this. Why is it in there? Who is the evolution of this Pseudomonas that has been evolving into this temperate zones across land? And so it is just kind of diverse, slowly shaped lichens that maybe didn't think of were like that's not what I thought lichen looked like. They are unique lichen. And they were able to, via the sequencing of this citrate transferase gene, CTS gene, they did some targeted sequencing and they were able to build the phylogeny of the different isolates that they identified in Iceland. And they were able to find the presence of some pathogenic Pseudomonas pathivars in there. And very similar evolutionarily, sequence-wise, to known epidemic infectious pathogenic strains, though not really seeming to cause a problem here in these lichen. So, yep. So this is they proved that pathogenicity was here. This lovely paper from Plant Pathology. They took it they extracted the syringe from the lichen, inoculated plants. They also took it from some. They did monocots, they did dicots, they did major agricultural crops, and yes, a lot of the strains that they found while living quite happily in this lichen had quite pathogenic effects here in the. So what's it doing there? That's where and they're now, so the wonderful Natalia is off on her postdoc looking at the metabolites of this. They're collecting more samples. They're collecting from other regions. They were out in Scotland. They're collecting more, how global is this? Is an all Peltigera lichen. When I came in, they had luckily sent out for sequencing and obtained whole genome sequencing of 16 of these syringe strains from their lichen samples. So I have the FASTQs and I'm ready to go. I have my pipeline at hand. I can do some QC. I can assemble, annotate, and compare the genomes. And I immediately thought this is a great opportunity for students as well. So I asked if it was okay if I involved my students back at Cal Poly. A handful of them said, yep, I'm in. I also asked if I could use some of my students who really enjoyed these galaxy exercises that we had been doing in class, and a few of them said, yes, I'm in. Because in Galaxy, we had assembled and annotated an E. coli genome. And I actually said, I don't mind if you still use that. Don't, right? So some of them are going to be running. We know it's unicycler, it's Proca, like I believe in the pipeline. But what it's giving them is students are claiming dibs to these genomes. We've set the parameters that everyone is to run these tools at. Some people are installing it on our own servers back home. That's great. They're interested in sort of more the infrastructure. I love that for them. The ones here are just running in their Galaxy instance, which you can do. It's been a long time / never since I've worked in microbes, and these small genomes are awesome. So they're going to actually use the same pipeline and compare the outcomes of their assemblies, where there's somewhere should be more worried about, etc. And then the fun part comes, and what we've been talking about with the team is I said this is like our, the 6th grade lesson on lichens was that algae or the cyanobacteria and the fungi. We know now it's more complicated than that. There are all sorts of other bacteria in there. Mutualistic, endolichenic bacteria is just a great phrase for it. There are a lot of other fungi. It's not just one fungus, it's multiple fungal. They are nematodes love living in there. Tardigrades love living in there. Is it just the house, or are there some benefit? Really interesting secondary metabolites. So our hypothesis, which we are testing, is that there may be some beneficial relationship between lichen, especially that genera, and Pseudomonas that should be identifiable viable via annotation of the genome and comparison to other phylogroups. So within R16, we know that some are pathogenic and some are not. What is common between R16, what is common between other non-pathogenic and other pathogenic. We're going to blow it up, go back to ABI and GenBank and grab a whole bunch of other Pseudomonas genomes, do big genome comparisons. But the types of things that they could be doing beneficially are things like nitrogen fixation, nutrient recycling. There's a lot of enzymatic activity in these bacteria. Pathogen defense, which is interesting because we were worried it was a pathogen, but maybe it launches a bit of an immune response that gets them on the defense. One I'm really curious about, and I said that 22-hour day, and we're up here very, kind of stark open spaces, is these bacteria are known to have pigment production capabilities. And is it something to do with actual maybe UV protection, genome damage protection, similar stress response genes that are being expressed by this bacteria that could help with temperature changes, like we certainly are seeing, desiccation changes, like we're seeing. And then other hypotheses out there, growth regulation, telling the fungi and the cyanobacteria who needs to be growing when and where, when is the growth phase, when is the photosynthetic phase. There have been some of these secondary metabolites seen from Pseudomonas potentially, and then some interest into maybe how they expand and colonize out. So this is giving Pseudomonas a real, it's there doing important, good job. And I think it'll be really fun to see if that pans out. It's been really interesting to talk to, especially at this university, this drive in the increase in biotechnology training in Iceland. They're really interested in, of course, for career opportunities, for resource management, right? We have geothermal here. They have geothermal here. They have hydroelectric here. How can we really improve biotechnology around those renewable resources? Fisheries is still one of the number one commercial spheres in Iceland. And so there's actually really interesting work. These are some students who are trying to synthesize bioalgae for food for fisheries. Maybe one of these, maybe we find something in these bacteria that's really good antioxidants, that's really good for the salmon, And then, of course, right, biomedical. It's really fun to see the innovation happening across the country. There is an alternative hypothesis, of course, there should be. And one is, the null hypothesis is it's just there for fun, for no good reason. And it's just random chance if it's there or not. And that's fine. They'll do more testing. They'll see if we don't find anything interesting, that's fine. And then there's this sort of more nefarious hypothesis. There's no beneficial, but potentially a concerning reason or rationale for their cohabitation. And not that it's bad for the lichen, but for this, and I did not know this till I moved here, feature of Pseudomonas, that they have a particular gene that when expressed, sole function is to be expressed on the outer membrane of this bacteria and serve as ice nucleating factors. And this is not new to anyone else maybe here. It was in the cover of 1976's Meteorological Society. So maybe it's new to biologists, I don't know. But you find a bacteria in the snowflake, and you've known that for a long time. What I didn't know is that that is actually a protein made by the bacteria in order to nucleate a snowflake in order to get around. That they use snow to disperse and to colonize, maybe. It's a hypothesis, but there's some good evidence of it, that the more pathogenic strains also seem to be having stronger evolution towards the production of these ice domain containing bacteria. And is this, we know that once it gets out there, it's really hard to contain. We know that Pseudomonas it is able to show up randomly on plants, that the outbreak seems sort of sudden and out of the blue. Are they using snowflakes as their hitchhiking the ride, as I said in my abstract, to go? So we're certainly, first thing I want them all to look at is, do we find evidence of ice nucleation genes in our strains? We know the sequences of those. They can do some nice pairwise alignments. They can look at, right, the pathogenic versus not pathogenic compared to outgroups. We can do that whole thing. And I know, we'll see. The jury is out. Choosing to do this via students, things are slower, right? I probably could have assembled all these genomes very quickly, but the training is why I'm here. The opportunity is why I'm here. And I think it's the students getting so invested in this project, both in San Luis and here, has been really, that's why I'm here. Why, does it matter? Well, agriculture, well, we think of Iceland as beautiful ponies and ice. Agriculture is critical in Iceland. It's an island. Otherwise, you have to bring it in cargo ships and in airplanes. They grow cucumbers and tomatoes and lettuce and a ton of barley. They grow really just like grass for their sheep and cows. And they really pride themselves on low pesticide use. There is not a ton of plant pathogens here. And therefore, and that has then resulted in a pretty low genetic diversity of the crops grown. There's like cucumber variant and tomato variant. And so letting Pseudomonas get out here could have really potentially devastating effects. And this ties into my last teaser in the abstract, is that everyone is aware that climate change is dramatically changing the landscape in Iceland. You can see it physically. Glaciers are shrinking and moving. Places that did not used to be habitable for crops now are. So this is our big global hypothesis, is that we worry about these Pseudomonas from Suriname using these ice nucleation genes and maybe now thawed out, maybe more, they never were in a region where they thawed out completely, and now they are, and they're able to enter into our water supply and become part of this process. And is that, should we worry about that here, especially in these vulnerable crops? If Pseudomonas comes out of these lichen and can get into the cucumbers and barley and et cetera, that they already showed it was potentially pathogenic for in that lovely study, that could be really serious bad news for the country. So this is all the questions tied together, and it's going to be wonderfully an ongoing collaboration. We will absolutely answer some questions. I'm here through December. I'm excited to take this back with us, though. It intersects genomics. We can sequence some more of these, bioinformatics, anything, right? Whole genome comparison, gene comparison, protein comparison, model those proteins, population ecology. How are these strains evolving? We're going to look for the evolution of these ice nucleating genes. Are there any of these pathogens we should be more concerned about? Then there's all this wonderful mapping and climatology and GIS and these glaciers moving around. So I think it's going to launch a really wonderful ongoing collaboration. It's also just been a delight to be here. I came here with my whole family. They are going to the local elementary school. They are speaking Icelandic at the local elementary school. My kids are learning. We are getting out and about and enjoying the northern lights and the ferns and all of the good stuff that Iceland has to offer. So if you do ever get the opportunity to visit, it is a beautiful country, and the north of Iceland is an especially special place. It's been very welcoming and friendly. And if you ever watched that Will Ferrell movie, it's perfect, the Eurovision movie. So I'm actually going to stop here, and I have some time for questions, and I'm happy to chat about any of this. Again, my email, if I can ever help with any of these conversations. And then here, I just ended up throwing my slides there because I know I mentioned a lot of papers and opportunities in the slides. So I want to thank again Scott and Arun for putting this together and hosting me. And I'm going to check my messages in the chat and open it up to any questions that might be out there as well. Ooh, fun. Yeah.
Thanks so much. That was great. Really appreciate that. Super interesting, incredibly interesting topic.
Isn't it fun?
I have a couple from students that I'd like to ask.
Please.
So one of them is Barbara's asking about microalgae being cultivated in Iceland, and is it grown there? How is it grown in large
Yes.
quantities? And is it grown
Yeah.
despite 22 hours of darkness? Like, is it only grown
That's right
in the
here.
summer?
Right. So it's there. This actually is a bioreactor incubator for algae for this fish food production. I know it kind of looks like servers or something, but maybe I don't know. It's actually an algae farm. So right. That's such a great question because yeah, the temperatures are so wildly variant. What will we be able to do outside and culture it in the fjords are freezing? I didn't know that fjords could freeze. I thought that was salt water. But yeah, it'll probably end up being in some sort of bioreactor, which is interesting to think about. You can really control like maybe a cocktail of different algae, really getting into one thing they're also doing with this is vitamin D is a huge challenge. Everyone struggles with vitamin D deficiency because of the winter, and they are bioengineering a lot of this algae cyanobacteria to have increased vitamin D capacity. And again, running these really big. Luckily, like electricity is not a problem. With the geothermal and the hydro, it's 90% covered by renewable resources. So why not run one of these really big farms here and use really clever bioengineering to make vitamins, make supplements, make fertilizers, all these things with these bioalgae? That's a great class. Great question.
Wyman is asking, is there a large computational difference when using Galaxy compared to
the real world
yourself? Like, do you see? I mean,
1,000%. Yeah.
Yeah,
it is. Absolutely. 100%. I'm not going to pretend. What I love about Galaxy is it takes real tools. So it's not some tutorial that like we built our own pipeline. They take real tools that are out there. Mother's there, bigwigs there, our friends are there. What they do well is they put a wrapper on it and they make it into a user interface that's not beautiful. Like if we made it now, we probably would make it more colorful and easier, but it's a user interface that is intuitive for anyone who uses the internet. So there's drag and drops, there's pull downs, but what they do nicely is they say like define this variable is a paired and single read, and define your variable how many base pairs, etc. So they do. I always think of like pseudo coding for running the tools. They use the right terminology, and then they always have a really nice readme of like here's what all the other parameters were that we chose not to put on here. And if you ever really, and here's where you go to, here's always has a GitHub link to the code, always has the reference to it. I take my students through that, and then I say, but we're trying to run an entire RNA-seq pipeline in a week. The dependencies and your different laptop, I've tried that before. I will never try to reconfigure your Windows 98 system to get the right, like, it lets us, so it's not programmatic and I think that's the part I miss and why I just added that sandbox.bio this year, is at least you get some of that wrangling and like moving files and saving files. But it's a beautiful user-friendly wrapper on important established computation tools, and you get 2 terabytes of data for free, which I've never come close to touching in my classes. So it's good for the practitioner.
Oh, that's really cool. So yeah, I had a question actually about, the thing I think about when I think of it's Pseudomonas, right?
Yeah.
Is biofilms. I always think about is biofilms. And the combination of biofilms and ice nucleation is very interesting. So another thing, for one thing, like having a biofilm with a potential protective mechanism might be interesting, but also the bugs don't care about the lichens, right? They care about themselves. Is ice nucleation possibly a defensive mechanism, right? Because if you have ice nucleation on the outside, it's possible that you can keep yourself from freezing because you can make yourself.
Because you keep the ice. Oh, you build your own little like ice house.
Is that any of that possible?
Yeah. Yeah, I don't see why we shouldn't look into it. Yeah.
And the other thing I might expect is
Yeah.
as they disperse throughout the world, I bet you they would lose isonucleation genes as well. Because the one thing you tell about evolution is that if you don't need it, it gets dumped real quick,
especially in bacteria. Yes, and I'm enjoying bacteria more. My poor lab's gonna like if it's there or if it's evolving to be there, like pseudogening, can we find like remnants of it, like, or the loss of it? Yeah, because it's under such evolutionary pressures, and it feels extra evolutionary up here. Like the pressures feel even more intense up here. And so if we're truly using it to, I love this. So yeah, there was a question in the chat. Would the bacteria provide insulation? And its point isn't the bacteria, it's that the nucleation of ice around the bacteria would actually protect the bacteria from internal, like the cytoplasm from freezing, which sounds, isn't the snow cold, but this is what like ice houses are. This is what it was known. If you actually surround yourself with ice, it's warmer inside. And so I'll have to look
it could help the lichen
into
too,
that.
right? Because the lichen, could be if the lichen select for those things, they might select them because they provide an ice nucleation event. So it could
because it is interesting. Yeah, because they're actually inside of the stromal hyphae. So they actually are like inside of there. So I wonder if it sort of even provides like insulation inside of the Jack. Okay, do you expect to find these ice nucleating genes on mobile genetic elements? Oh, my microbiology. And yes, so there is, right. We're definitely going to look at horizontal gene transfer. We want to look at about how these are sharing. Is it just a Pseudomonas thing? Are there other bacteria? I haven't gone into that too much, but that's a great question we should ask about. Is it just Pseudomonas? I don't think it's just Pseudomonas, but I don't know how expansive that has become. And then is there, horizontal gene transfer between isolates of things living inside of the lichen? We're interested in also culturing up other stuff inside the lichen, when they wasn't just the cyanobacteria, and that like it's more busy in there than we thought. Is there some interesting shuffling of genes inside of that lichen ecosystem as well? Cool. So great. I would recommend the chat. Arun put some wonderful links. Yes, I will absolutely share the class. I'm saving everything and making it really open. Great question. Little family who's interested in doing a Fulbright. How was the transition? Great and weird. I mean, we're from Southern California, and do we have hard, dark days? Absolutely. I went on a walk today with my daughter in the snow, and she was trying so hard to articulate it. And I was like, there's no word. I get what you're trying to say, but I miss people, but I'm so happy, but I'm sad that it's almost over, but I'm happy that I get to go home and all the things. My only comments are children are just the most resilient people I've ever, like, we dropped him in this brand new school, not speaking the language, not eating the food. Here's your lamb stew for lunch and your fermented fish snacks. And that part I need to not worry about. Like, and almost me trying, like, if I was to be anxious, it would have imposed that on them. This is what we're doing. This is our family adventure. We framed it as like, we try interesting new things. You guys are great. And they're made friends. We got into the sports. I will say being in a smaller town, I think was really beneficial. Like not being in Reykjavik, but being in this little town, people were like, we were celebrities. People were so excited that we were here. And at school, there were celebrities. Do we miss things? Yes. Do we miss our home? Yes. But now with Zooms and calls, like I think they don't feel as disconnected as probably they would before. I highly recommend it. We're at the right age. My kids are 6, 8, and 11. And I think that was kind of a nice age. The teenager's not too teenagey. The younger one's old enough to carry his own backpack. Like it felt like a right time for it. But I wanted to do it again. I think that way it opened their eyes. The way to be the other for a while, to not speak the language, to feel what that felt like to be the other, even though like they'll take that forever. Walking to school today in the snow, they'll take that. Academically, people are worried they're behind. Like this is so much more than that. Elementary school is great and wonderful and bless them, but it's all about learning and yeah. So highly recommend. Please reach out if you have any questions about the Fulbright process, about families. I love it.
Now don't reach out to me. They denied me.
reach out to me. Did you
No,
apply to the targeted one?
I actually got approved for going to Denmark. They loved my proposal, and then the State Department just said no. They just said
I've
no.
never heard.
Yeah, they said no,
Whoa,
and
you
they
have some
didn't tell me
shady
why.
background.
Yeah,
Oh, I
they
always
tell me
heard as
what
soon as
about.
you got the country's approval, you were pretty like.
Yeah, I mean they.
Don't
I don't
know.
know what happened, but they just said
Maybe it
no.
was funding or
They don't tell
something.
you. They don't
I'm
actually tell
sorry.
you why.
Yeah, it's
But
not a great product. Like
you did it by going to an RP, like our RFP, and so they're
kind
really.
of. It was through the phone. Yeah, they were really looking for someone, and so that felt like as soon as I started talking to them, and as soon as they wrote me that letter from here to like, it felt great.
I wrote a killer proposal. I wrote a killer proposal.
Right,
it was the best proposal I'd written in a long time, and the people loved it. The scientists loved it. So, but
weird.
Anyway,
I'm sorry.
It's okay.
All right, there's a question about plans to compare gene expression from the Stingray. Yes. So we haven't yet. So we do RNA-seq between this pathogenicity. We haven't. We absolutely should. I think that's a great ongoing collaboration. I'm sure someone's done I'm sure there's a transcriptome for Pseudomonas, but I would love to. Are they expressing these genes? That's always the next question. The genome is great, but are they actually expressing these nucleation genes, these pathogenicity genes? We need to ask that. I'm curious if there's something about the plant environment versus lichen environment. Yeah, right. I don't know, right? Is there something about, and why just this genera of lichen? There's 800 different species. There's like 10, 20 different genera. Why just this genera with this weird leafy? It's really spongy. Is it I know it's so funny that Scott said they're out for themselves, but I was like, they're helping out. It's commensal, but maybe that's just a really nice spongy host that keeps you safe in the winter. And that could be the reason too. My email address, yes. Oh, perfect. Thank you. It is jdavid06 here at the end. I'd love to chat about anything.
We had a couple of questions from the
chat. Sure.
There's still some time, so that's good. So
please, yeah.
One is a practical one. Aaron asking, do you have some favorite tools for identifying pathogenic bacterial genes? Do you just use like the standard like annotation, or do you have a specific pathogen gene, viral gene annotator?
Okay. Have you heard about BugSeq yet? I think you know. No, they're in like Canada. Anyways, yes. So we look at databases. It always comes back to database, and BugSeq has put together a really nice pathogenicity database. Sort of feels like dbSNP of the bacterial world. I don't know how broadly expansive it is, but it does a nice job of, there's a database you can get. There is actually a pipeline that they have as well. They're really generous with academics. They're trying to get it into like clinical use, so there's like hospital costs, but a really wonderful company that is trying to categorize all of this. All of these pathogenic variants. Again, human focused right now, but I think they're understanding that plant pathogenicity is a thing, other viral pathogenicities are a thing, and we need our ClinVar. We need the real database that we can query against. So I was going to go with BugSeq. I bet there's other ones out there. It's a pretty cool company. They're great folks.
One other question was: did you find any unique bacterial proteins or enzymes that allow them to survive these low temperatures? I mean, I remember these beetles I had once actually produced antifreeze.
Right.
Was there anything like that?
Yeah. So that's what Natalia is. She did a whole metabolome profiling of them. And we certainly know that these lichens, especially these ones up here, are making fascinating metabolites, interesting defense mechanisms, interesting UV protection mechanisms. And so she's really going after the metabolic. And then, of course, so like we should absolutely do we should do the transcriptome and find it right in the middle after different, in the full daylight, in the full darkness, in the freezing, and the not, and see what metabolites are being turned on. it comes back to my why I fell in love with plant biochemistry is they only have their genome. If there's drought, if there's stress, if there's sunshine, like they only have their genome to come back to. And so their solution to these problems is going to be somewhere in this genome. And can we find it? And then can we maybe hijack these metabolites and thinking about them for biotechnology? But yeah, antifreeze, what do you do when you are a cyanobacteria is dark for 22 hours a day? Like there's going to be some interesting nutrient cycling going on. Like there's going to be some interesting, there are ones that utilize sulfur instead of nitrogen. Like they are wildly creative because of this really intense evolution up here.
This sounds like an absolute experimental thing. If you can,
isn't it fun?
It's like say, I'm going to dump some UV on these guys, see if they produce something crazy, or I'm going to freeze them.
Yeah, freeze them.
Whatever.
Step on them. yeah. And it's such a cool little system. Like this is, I just feel like we're going to peek into this community and what we can understand about these relationships and the gene expression and like it, but it's all in this little microcosm of a community in there. And maybe I'll get to come back and stomp around and collect some of them.
Amazing. Let's see.
Awesome.
All right. Well, I mean, we are getting on towards one.
Yeah.
And there's a lot of questions. I don't want to keep you forever. Everyone's so excited to.
Awesome. Please, and if you want to help out, I mean, this is a globally collaborative. I am open for other folks jumping in on this, especially microbiologists who knows what they're talking about. This is a fun ongoing collaboration. They are very keen on like sharing this. They understand that like it's a small university here. They would love people to be involved on it. I can't emphasize enough.
That's all I'm saying. Look at those biofilms.
Yeah. Look at those biofilms. All right. I like it.
The Pseudomonas are like a mass. For those of you who are on this, those are massive problems with cystic fibrosis. And then you find them all over the place. Like I know daycares, when I studied daycares, they grew all over the surfaces. I think if you just give them a little food, they'll make a little slime everywhere you go. Whoa. And so I can't believe that they're not, and what's in the biofilm is really fascinating because sometimes they actually exude DNA into the biofilm. They exude proteins and DNA as a matrix, and biofilms are wild, absolutely wild, and they're just understudied for bacteria. But Pseudomonas are like the biofilm.
Okay.
I want to think we'll look at enrichment of like what is biofilms that are on
what's there.
The lab,
because I think it's a community. I don't think it's just these. Yeah, it's definitely not just these guys. And very cool. And what that protective or defense or selfish reasons are there. There's got to be something. Yeah. Very cool. Super grateful for the colleagues here for sharing this work, all this like groundwork that they lay. I feel like I just got this like package that I have a million questions. So yeah.
No, please, when you get the chance, do share like the curriculum and stuff like, because I think especially I've used Galaxy for teaching undergrad level bioinformatics classes, and I think it's such a huge boost because I think people think it's some ivory tower, right, where it's so difficult to do because they don't know how to do any coding. But then I think it just makes bioinformatics and genomics that much more accessible.
And then if you like that tool, then we'll go and install it and do all the stuff. But if you just want to try it and run your one E. coli, get in there, go for it.
It works. And we've been doing some developing for Galaxy too, honestly, like some of the more complex sort of pipelines, you can, our goal was to sort of make it much more accessible, right? Because I think there's still that huge learning curve when it comes to bioinformatics tools.
And we need all of our students to be functionally aware of what's out there. They don't all need to be programmers, but they need to know that that's something they can access, that it's something they should think about, collaborate with. And just once you do it yourself, you're like, whoa, that is so powerful. Yeah.
I agree.
Yeah.
All righty. Well, it's past one. So I want to thank Dr. Davidson for doing this and, wishing you all the best.
Yeah, go look for some northern lights. Yeah.
Awesome.
Get out there.
You're going to go.
Next week, I'm going to be the speaker. So I don't know, Scott, you can introduce me maybe, or I can just introduce myself. But I actually have a plan. And my goal was to not talk about my own research, but actually talk about 4 of my super women PhD students and their research. And so we're going to be taking some pictures in our lab meeting today of all my 4 superwomen PhD students. And so I'm going to talk about their research. And so it'll be really fun. Some of it is heavy on the math, some of it's heavy on the coding. Other things are a little lighter on the coding and a little bit more analytical, but it should be fun. So that's what I'm going to do. So I look forward to seeing a lot of you attendees there next week as well. Again, same thing. Pacific noon. We'll see you next Friday. So
send me your CV.
Thank you.
I will. All right. All right.
Thanks, everyone.
Really
good to see you. Bye.
Bye.
Bye.